My dear children, you are all warmly welcome to our Vidya Lakshmi YouTube channel. I am Mrs. Lata from Botany Department. So today we are going to learn the first chapter in the 12th standard Botany. Okay, what is the first chapter? It's a reproduction in plants. Shall we enter to the class now? So reproduction in plants. So in this chapter we are going to learn that is the following subtopics. The first one is asexual reproduction and the types of asexual reproduction and types of the vegetative reproduction, conventional methods. Okay, that's a cutting, grafting and layering we are going to study today. So what is the reproductions? Okay, so reproduction is a biological process, so by which a new individual organisms are formed from their parents, isn't it? The reproduction is a vital process for the existence of a species and it is also brings suitable changes through variations. Okay, so in the offsprings. So through sexual reproduction they can bring some variations in the offsprings. Okay, but in the asexual reproductions they cannot bring any variations in the offsprings because the offsprings of asexual reproduction that is identical to their parents. Okay, that is not differ from their parents. So what is the definition of reproduction? Reproduction is a vital process for existence of the species. They need to survive. So continuously, it also brings suitable changes through variations in the offsprings for the survival on earth. Okay, so reproduction is very important for that evolution. So what is the evolution? So the organism having the continuous changes in several years. Okay, that is known as evolutions. So what are the types of reproduction? Actually, it is divided into two. One is asexual reproductions and another one is sexual reproductions. Now you can see some pictures. There is asexual reproductions in bacteria. There is a binary fission method and that is in the amoeba. Okay, there is asexual reproductions. Type of asexual reproduction is occur in the amoeba and bacteria. So what about in the higher plants? That is here in flower. Okay, so then higher plants it has a sexual reproductions. Okay, the flower is the site of sexual reproductions. Okay children, shall we enter to the next topic? That's a subtopic. Today we are going to see asexual reproduction alone. Okay, so what are the characters of asexual reproduction and what is the definition of asexual reproductions? We will see that. So what is the characters? So what are the characters of asexual reproductions? In the asexual reproduction, a single parent is involved. Okay, so only one parent is enough for that. So that is a single parent. That's a uniparental in condition. This is called a uniparental. So only one parent is enough. Okay, the gametes are not formed because there is no meiosis. The division meiosis is not takes place here. So the gametes are not formed. Male and female gametes are not formed. So there is no fertilization. So no fertilization in adakar. Yeah. So there is only mitotic cell division is occur. Okay, through the mitotic cell division which gives rise to the young plants. Okay, that's a mitotic cell divisions. Daughter organism genetically identical to their parents because they have only the mitotic divisions. Meiotic division nadanda kandipa daughter cells la eppadi irukum differ from their parents, but here the genetically identical parents are formed because they have mitotic division is occur. Okay, multiplication occurs rapidly. Okay, so this is very rapidly multiplication. Then only they have to increase the size of the organisms. Okay, increase the tissues and organs. Okay, so these are all the characters of asexual reproductions. The first one, the single parent is involved, that is uniparental in conditions. The gametes are not formed here, so there is no fertilizations and only mitotic cell division is occur, so there is no meiosis. Okay, the daughter organisms are that is exactly so like their parents and multiplication is occurs rapidly. So these are all the characters of asexual reproduction. So what is the definition of asexual reproductions? So what is the definition of sexual reproduction? The sexual reproductions means the male and female gametes are fused together to form a zygote, isn't it? The male and female gametes are involved in the process in the sexual reproduction. But what about in the asexual reproductions? The male and female gametes are not involved. Okay, so without the involvement of the male and female gametes, without the involvement of the gametes. Okay, so that is the asexual reproduction. So asexual reproduction is a method which helps to perpetuate. What is the meaning of perpetuate? That is a continuous, okay, continuity. It is own species without the involvement of the gamete is referred to as a asexual reproductions. Okay, continuously they have to help the young ones. Okay, they can continuously produce the young ones. That's a without involvement of the male and female gametes. Okay, because they can produce by the mitotic cell divisions. So what about the asexual reproduction is occur in some algae, fungi, monera and protista. So that is occur in the lower organisms in the plants. Okay, so what is the clones? As a result of asexual reproduction, the offsprings are similar to their parents, okay, exactly identical to their parents, that is morphologically and genetically identical to the parents. So that is known as a clones. So we can see the clones in the bryophyllum. Okay, in the margin of the bryophyllum, so we can see there are several buds. Okay, each and every buds they can give rise to a entire plant when we have to separate the buds from the leaf. Okay, so this is identical to each other. Each and every one is similar to each other. Okay, identical to each other. Okay, that's a correct term, is identical to each other. That's a morphologically and genetically identical. So that buds are known as a clones. This is a clones. So we studied definition of the asexual reproduction and what is clones. What are the examples for asexual reproductions? So what are the types of asexual reproductions? Okay, so the types of asexual reproduction, the first one is the conidia. Okay, so conidia is nothing, that is a spores. Not only in conidia, zoospores, aplanospores, all type of spores, they can reproduce by asexual method. Okay, next one is a budding, and another one that is a fragmentations, and another one is a jamme, and next regeneration and binary fissions. So these are all the types of asexual reproduction. Okay, so here that examples are very important for your examination point of view. Okay, so the conidia, what are the examples in the conidia? That is Aspergillus and Penicillium. Okay, so what are the examples for the budding? That is a yeast and hydra. What is the example for fragmentation? That is a Spirogyra, that is belongs to algae, isn't it? So gemme, that is a Marchantia, that is in the bryophytes. Okay, Marchantia belongs to bryophytes. And regenerations, that's a planaria. Okay, so regeneration is they have to regenerate the damaged parts or missing parts. Okay, so example for in our body, that is a hairs and nails, or so that is a regenerated. Okay, and binary fission, this is a common method, common asexual method in the bacteria, that is a binary fission. So all the examples are very important for the examination point of view. Conidia, Aspergillus and Penicillium, budding yeast and hydra, fragmentation is Spirogyra, and gemme, that is a Marchantia, another one is a regeneration planaria, and binary fission, that is bacteria. So next, what is conidia? So conidia is a type of spores, actually it's a thick-walled spores, okay, thick-walled non-motile spores. Non-motile spores means they don't have a flagella, okay, that's a non-motile spores. That is a picture of Penicillium, Aspergillus and lichens are there, okay. So this is the conidia. The conidia are arranged on the conidiophore. So conidias are arranged on the conidiophores. So this is a conidiophores that is arranged as a chain-like structures. Okay, the conidia eppadi arranged aayirukku, or chain mari arranged aayirukku, arranged as a chain-like structure. That's one above the other. All the conidias are arranged. Okay, so the conidias are attached to their stalk-like structure. This is known as a conidiophores. This is called a conidiophores. So during the maturity, it is liberated from the conidiophore. That is a separated from the conidiophores. Okay, the spores are undergoing the germinations. During the favorable condition, the spores are undergoing the germination, and which gives rise to the hyphae. Okay, the hyphae is together to forming a plant body of, that is a Penicillium or in the Aspergillus. Okay, so this is a conidia, a type of asexual reproduction. And next we are going to see the budding. Okay, that is here. So, budding is a small bud-like structure is present on the parental plant body. Okay, it may be present in the base of the plant body. We can see a bud in this region, that is in the hydra. Hydra in the base of the hydra we can see a small bud-like structure. Okay, when that is attained maturity, okay, after the maturation, that is detached from the parental body and which gives rise to the entire plant. So, that is a budding method. We can see the budding method in the yeast and another one in the hydra. Okay. So, in the hydra a small bud-like structure is present. So, after the maturation it is detached from that. Okay. So, budding is also a type of asexual reproduction. And next one is a fragmentation. Okay, what is a fragmentation? A small piece of any plant body we consider as a fragmentation. So, actually it is the process, the breaking down of parental plant body. Okay, so just you have to take the parental plant body. This is an example of Spirogyra, that is a algae, it's a hydrophytic organisms. Okay, a filament of the algae, just you have to take a filament of the algae and break it into two small pieces or two fragmentations. Okay, so each one gives rise to a entire organisms because that is undergoing the mitotic division to form a entire organism. That is the fragmentation. The fragmentation is also a type of asexual reproduction. And next one is a gemma. Okay, so gemma is nothing, that is a small cup-like structure that is present on the plant body. Okay, that is in the thallus. That is a Marchantia. Marchantia belongs to the bryophytes. Okay, so in the plant body of the Marchantia, we can see a small cup-like structure. Okay, that cup-like structure is called, that is here, gemma. Okay, so this is a gemma is a plural formation, this is a gemma. Yeah. Each gemma consists of a small bud-like structure which is undergoing the mitotic divisions and they have to attain the maturity. Okay, just you have to detach from that parental body which undergoing the germination and gives rise to the entire thallus. Okay, that is in the Marchantia. So, what is the asexual reproductive part in the Marchantia? That is here gemmae. Okay. So, that is present in the liverworts and mosses. And next one is here regenerations. Okay, so what are the parts are regenerated in our body? That's a hairs and nails, isn't it? So, regeneration is a natural process. Here, that's a replacing or restoring the damaged or missing parts. Okay, so it may be a cells or tissues or organs or even entire body parts to full functions in the plants and animals. You can see here, this is a planaria. So, a single planaria is divided into two. Okay, a single planaria is divided into two. So, this is a single which is divided into two and each one is act as a new organism because each one they have to restoring the damaged parts. Okay, so even that entire parts, okay, entire body parts they can regenerate. Adarkude ella body parts regenerated panni oru pudu organism adu function pannum. Okay, new organism individual adu function pannum. That is one of the example is a planaria. Okay, so what is a planaria? What is a regenerations? That is, a regeneration is a natural process of replacing, restoring damaged or missing cells, tissues or organ, even the entire parts of the body parts, even entire body parts they can regenerated once again. Okay, so that is a regeneration. And next one is a binary fissions. That is very we know everybody we know very well about the binary fission. The binary fission is a common method in the bacteria. Okay, it's a type of asexual method. Okay, that is a sexually is that is they have done by that is a conjugation method. Okay, sexual method na conjugation method. This is asexually reproduced by the binary fission method. Okay, so here a single bacteria so divided into two daughter cells and each daughter cell is act as a individual organism. They can lead their life independently. So that is the binary fission. So first the nuclear materials is divided here. Okay, so this is the nuclear materials. The nuclear materials are first divides. The nuclear material is here first divide and followed by a median constriction is occur in the wall. The median constriction is occur in the wall. Okay, so finally it is divided into two cells. Okay, so followed by the cell wall is divided into two. So that is a binary fission method. அப்போ first வந்து nuclear material divide ஆகுது, அதை follow பண்ணி bacterial cell wall வந்து ரெண்டா divide ஆகுது. That's a simple median constrictions வந்து, that is we can see a small median constrictions. Okay, we can see the median constrictions. So next one is your vegetative reproductions. Already we studied, that is your asexually reproduced method. Okay, asexually reproductions. Okay, now we are going to see the vegetative reproductions. What is the vegetative reproductions? The vegetative parts are involved in the process. So what are the vegetative parts? That's a root, stem and leaves. That is considered as a vegetative parts. Okay, just you have to take the vegetative parts from the plant body and keep it in some other places in the suitable medium, which gives rise to an entire plant. So that is the vegetative reproductions. Okay, so vegetative reproduction is also a form of asexual reproduction in which a bud grows and develop into a new plant. So which is considered as a bud. Okay, so in the bud நம்ம சொல்றது வந்து எதை சொல்றோம் அப்படினா, so it may be a root or stem or leaf. Okay, it may be a root or stem and leaf, that is grows and develop into a new plant. Okay, it's not only used for vegetative propagation, this is also used for storage. Okay, இதுவும் storage பண்றதுக்கும் யூஸ் ஆகுது, and perinations. Okay, storage means they have to store the food materials, even starch. Okay, so that is a tannin, and some of the materials they can store inside. So example for in the cortical cells, the cortex, pith, they can store the food materials. Okay, the perinations அப்படிங்கிறது, they can, the organism able to survive from one germinating season to another germinating seasons. Okay, so during the favorable conditions we can see the shoot, that is here aerial regions consists of shoot system. Okay, during the unfavorable conditions, so we cannot see that aerial shoot, but that is survive in the buried, they survive in the soil. That is unfavorable conditions la enna pannu appadina, that is survive in a particular regions. If example வந்து soil குள்ள வந்து underground குள்ள அது வந்து survey ஆகிட்டு இருக்கும். So during the favorable condition it will germinate and gives rise to a shoot system. Okay, that is a perinations means the organism, the ability of the organism, they can survive from one germinating season to another germinating season. Okay, ma? So what is the propagules or diaspores? This is very important one mark questions. Okay, so what is the diaspores and reproductive propagules? Okay, so diaspores and reproductive propagules are nothing, any unit of the reproductive structure. It may be a root or stem or leaf. Okay, any unit of reproductive structure used in propagation is called a reproductive propagules, or this is called a diaspores. Okay, so we can see the reproductive propagules in the tomato, in the potato. This is that a small eye spot. Okay, in the eye spot adha nam enna solrom, this is a eye spot or considered as a propagules, or that is a diaspores. Okay, and here that is a buds. In the bryophyllum we can see the marginal buds. Okay, the buds are located in the notches. Okay, and the marginal irukkudiya notches la vandu irukku. So here the buds are arised from the node. Here the buds are arised on the node. Okay, so these are all the reproductive structures, reproductive unit, that is considered as a reproductive propagules or diaspores. Okay, it's very important one mark. And what is the function of that is a vegetative parts? That is not only used for the vegetative propagation, they can storage, undergoing the storage and perinations. And next, vegetative reproductions in the root. Okay, so the root of some plants, they develop vegetative or adventitious buds. Okay, that are from the adventitious buds, the aerial shoots are formed. Okay, so the roots are act as a vegetative propagules. Example for in the Morea, Dalbergia and Millingtonia. So these are all the example for the vegetative reproduction in the root. Okay, Morea, Dalbergia and Millingtonia. Name of the species are very important for the one mark question. Okay, so here the some tuberous, not only it's involved in the propagations, and some of the root tubers, they can store the food materials. That is example for, that is a sugar beet, that's a Ipomoea batatas. Okay, Ipomoea batatas are not stem tuber, this is your root tubers. Okay, so roots apart from the developing buds, they also store the food. Example Ipomoea batatas and dahlia. Okay, some tubers, the adventitious roots or are raised. Okay, so the adventitious roots are gives rise to the entire plant body. Okay, some tuberous adventitious roots. Apart from that, developing buds also is there. Okay, that is store the food materials. Example for storage in the root modification, that is Ipomia batatus and Dahlia. And next we are going to see that vegetative reproduction in stem. Okay, here we are going to so many types of vegetative reproductions. Okay, what are the parts are involved in the vegetative reproduction in stem? Okay, the stems are used for the vegetative propagations. The first one is underground stems. Okay, what are the underground stems are used for vegetative propagation? The first one is a bulb, you know very well, that is Allium cepa. Okay, so that's a liliums or that is belongs to the bulb. Okay, another one is a tuber, that's a Solanum tuberosum. And next one is a rhizome. What are the example given in that? So, Gingiber officinale, Musa paradisiaca and Curcuma longa. So, these are all examples for rhizome. And another one is a corm. Corm, that is a Amorphophallus, that is a Colocasia. Okay, Amorphophallus and Colocasia, these are all the example for the corm. Okay, that is the first one is a bulb, tuber, rhizome and corm. So, these are all underground stem modifications. Okay, so this underground stems that is used for the vegetative propagations. And some of the subaerial stems, the subaerial stems also used for the propagations, vegetative propagation, that is your runner and stolon, offset and sucker. So, what are the example for runner? Centella asiatica, stolon mentha and fragaria. Offset, that is Eichhornia, and another one that is a sucker, is a chrysanthemum. Okay, so these are all the vegetative parts in the subaerial stem. Okay, so what we will see that. So what is the bulbs? So the bulbs are, that is a modified shoots, that's an underground part. Okay, the bulbs are modified shoots bearing apical and axillary buds. Okay, so in the bulbs vandu ennada maarudha, that is a modified shoots. What is the shoots? That's the aerial part is considered as a shoot. Okay, so here that is a apical buds or gives rise to the shoots. Even apical bud, that means that is a terminal buds. Okay, another one is a axillary buds. The both buds are arised from the bulb. Okay, that is here. So modified shoots, they can give rise to the modified shoot. Entire shoot system on the form agru. So next we are going to see that. So underground stems, that is a tuber. Okay, that is example for that is a potato. Okay, we can see the eye spot in these regions. Okay, the eye spot is the axillary part. Okay, this is the axillary region. From the axillary regions, the shoots are arise. The tuber is a modified, is a very thick and a swollen stem. Okay, the potato is here, tuber is modified stem. Actually, this is a stem. The stem regions usually linear, but this is not like that. It's a modified, is a swollen, thick stem. It consists of rich starch is there. Okay, so it bears buds. Buds arise, that is your eyes. Eye spot mari irukku and the region la irundha buds are raised. The buds are raised from the eye spot, that is axillary bud will give rise to a shoot system. And next is the underground stem. Another underground stem is a rhizome. Okay, so rhizome is also a underground stem. The having the buds, nodes and internodes are there. Okay, adirukku kudiya regions appdin paathana, so we can see the nodes, internodes and buds are there. Okay, so this is the nodal region. Okay, this is nodal, this is a internode. This region is a internode. This is internode. Okay, and this is the axillary buds. Okay, so that from the axillary regions, that is the buds are arise. Where is the axillary regions? Actually, the regions between the node and internode. So this is the one node, this is the another node. So between this region, that axillary buds are arise. Axillary buds on the form of. Okay, so that is the buds are arised from the internode or nodal region, which gives rise to the aerial shoot. Okay, this is also example for the vegetative propagations. Actually, vegetative propagations from the underground stems. So next one is a corms. Okay, so corms is also underground stem, that is gives rise to a entire shoot system from the axillary region. The buds are arised from the axillary regions. The corms are covered by the scaly leaves. Okay, the corm bears the buds. Okay, they can give the buds. The buds are, that is gives rise to the shoot system. Sprouting means there is nothing that is undergoing the germination and gives rise to the new plants. New plants will arise from the buds region. And next, subaerial stems. Okay, subaerial stems, that is your first one is a runner. Okay, so runner is nothing. Here we can see the plant, that's a Centella asiatica. The Centella asiatica is spread over on the soil, that is the surface of the soil, isn't it? So this is your runners or specialized narrow green branches. And the branches it's a green in color. Okay, it's a specialized, this is a narrow kurigi irukkudiya or green horizontal branches. Okay, so we can see the adventitious buds, adventitious roots are arised from the crown. Okay, in the crown keela da adventitious roots vandu arised. Just you have to cut it from that parental plant. Okay, and keep it in some other places in the suitable medium which grows into a new plant. Okay, upon the develop at the base of the roots. Enga develop agudhu appadina the roots vandu base of the crown. The crown, this is a crown, the shoot system. So idukku keela da the roots vandu form agudhu. Okay, just you have to separate the part of the plant and keep it in some other places. Okay. And next one is a stolon. Stolon is also that's similar to that is a runner, but it's a creeping horizontal branches. Okay, so that is also gives rise to your, that is your branches from the buds. And next one we are going to see the subaerial stem. Subaerial stem, that is the example succus. Okay, succus, the best example for succus, that's a chrysanthemum. Okay, the suckers are arise from the base of the erect shoots. Okay, and the erect shoots mela irukum, adukku base la it is formed. That is the shoots that suckers means at the adventitious buds or adventitious roots are formed from that. Okay, and after some time, so after the 1 or 2 days, just you have to detach the, so that's a daughter plant from the parental plant. Okay, each daughter plant which is act as a new individual. Okay, the subaerial stem sucker. Next, another one we are going to see the offset. Offset is very important part. Okay, so it is a very important one mark in the offset. That is here, it has here only one node is there. We can see only one node. So can you give the example for the one node subaerial stems, that is the offset. Okay, this is the only, so one nodes are, so one internode, not one node, this is one node, this is another node. So in between that is only one internode is there. Okay, so only one internode length is there. In base of the crown we can see that is the adventitious roots are arised. Okay, this is aquatic plants. So that's it. Just you have to break a part. Okay, one internode irukkudiya and part vandu enna panna, just you have to break it and separate it from the mother plant and which is keep it in some other places. Okay, a suitable mediathula water la potutom appadina it will grow as a entire plant. That is a subaerial stems example offset. And next we are going to see the vegetative reproduction in leaf. Okay, we know very well that's a bryophyllum. So bryophyllum is the best example for the vegetative reproduction in leaf. Okay, in the margin of the bryophyllum we can see the small buds of the leaf. Okay, small buds are arranged on the margin of the bryophyllum. In the each notches we can see the buds. Okay, each and every buds are detached from the parental leaf which gives rise to the entire plant body. Okay, so actually the buds consist of adventitious roots. Okay, each and every buds around the bryophyllum leaf, it consists of adventitious roots. Okay, so they can give rise to entire plant body. In some plants, adventitious buds are developed on the leaves. Okay, that is known as so epiphyllous leaves. What is epi? Epi means that is attached. Phyllis means leaves. Okay, some of the buds are attached to the leaf. So that is known as epiphyllous leaves. So when they are detached from the parent body, so they grow into a new individual. Okay, example bryophyllum, Scilla and begonia. Okay, so in the begonia, this is a begonia plant, this is a Croton's like structure. The leaves are crotons leaves. Okay, leaves on the crotons. So when the leaves are injured, okay, so they can give rise to the entire plant body when which is contact with the soil. Otherwise, just you have to take a leaf from the plant body and keep it in the soil. So it will grow as an entire plant. Okay, it's an amazing thing. It's a begonia. And next one is your Scilla. Okay, in the Scilla is a narrow, is a foliage, long foliage leaves are there. In tip of the foliage leaves, we can see the adventitious buds, adventitious roots are arise. Okay, just you have to detach from that, detach from the parental plant and keep it in some other places. Okay, you have to planting in some other places, it will grow as an entire plant. So this is also the example for vegetative reproduction in leaf. So it's another thing. So a leaf part which gives rise to an entire plant body, that is considered as a vegetative reproduction. So next we are going to see the bryophyllum. Already I explained about the bryophyllum. So the bryophyllum consists of a small buds. So all the buds are raised so around the leaf. Okay, this region is considered as a notch. The region kenna peru? This is a notch. Okay, so each notch we can see the buds, that is a clones. Okay, all the buds are so similar to, that's identical to each other. So in that notch, in that buds we can see a small adventitious roots are arised. Okay, in the bud keela or adventitious roots varum, just we have to take a small bud, or bud mattu eduthu or place la vachittingna, it will give rise to your entire plant body. Okay, that's a vegetative reproduction in a leaf. It's a succulent. The succulent it is store the food materials. Okay, so they can store the water. Okay, enrich water they can store inside the leaves. That is a succulent. And notched on the ends. Notched, this is called a notched. Okay, adventitious buds develop at these notches. Okay, and this is called epiphyllous buds. Okay, epi na attached, phyllis na leaves. Okay, so leaves ku mela or buds vandu attached aayirukku. So this is known as a epiphyllous buds. So another one is a Scilla. This is a picture of a Scilla. Okay, so a Scilla has is a narrow. This is what type of leaves? This is a foliage leaves. It's looking green in color. So this is known as a foliage leaves. So foliage, they can undergoing the photosynthesis. They can do the photosynthesis. Okay, this is a foliage leaves. The foliage leaves are, this is a long and narrow. So narrow. It's not very broad. Broada kadiyadu, narrow. So actually this is a long and narrow leaves consists of a buds. In the tip the buds. Okay, the base of the buds we can see the adventitious roots are arise. Base of the buds adventitious roots are arise. Okay, so this part is able to give a entire plant body. Okay, this is a vegetative reproduction in leaf. So it's a narrow epiphyllous at the tips where which is present, the buds are present in the tip of the leaves. Okay, what about in the bryophyllum? The buds are arranged, that is arranged on the notched regions. Okay, but inga vandu enga arranged aayirukna, that is a tip of the leaf. Okay, these buds are developed into new plants when they touch the soil. Okay, so otherwise they cannot give rise to the entire plant body. So all the buds, they must touch the soil, they must contact with the soil, then only they can give rise to the entire plant body. Okay. So next we are going to see the bulbils. Okay, bulbils, there's a bulbils is a small bud-like structure that is on the plant body. Okay, just you have to take a bud and keep it in that some other places. Okay, they can give rise to a entire plant body. Okay, that is undergoing the mitotic division and which gives rise to the entire plant body. So that is known as a bulbils. Okay, so the bulbils also a type of vegetative reproductions. Okay, they are produced in the axil of the foliage leaves. Okay, axillary regions la in the bud small structure on the produce. So what is the axillary region? The region between the node and internode. Okay, so this region is a axillary region. In the axillary region we can Dioscorea. The name of the plant is Dioscorea. So we can see a small bud. Okay, a fruit-like structure, a bud-like structure is formed. Okay, in the place of axillary bud. The floral buds can be modified into bulbils. And the floral buds are bulbils modified. Then it grow into a new plants. Okay. What about in the agave? Agave consists of so that is a floral buds. In the center of the floral buds. The floral buds are able to give rise to the entire plant body when they have to contact the soil. Okay. So there is a bulbils also, that is a type of vegetative reproductions. Okay. Next, last one is biturions. Okay. So here the special type of fleshy floral buds are there. Okay. There's a floral buds are there, that is in the aquatic plant. So they are swollen and reserve the food material. It's a swollen, reserve the food material. It's look like a tubus. Okay. So swollen food material store pannom. But during the spring, it forms a new plant. Okay. Spring season, they have to give you a new plant. That's a Potamogeton and Utricularia. Okay. So Utricularia is also the insectivorous plant, isn't it? So they can give rise to the entire plant body during the spring. Actually, floral buds are gives rise to the entire plant body. So next we are going to see, that is what are the advantages and disadvantages of natural vegetative reproductions. Okay. So we know very well this is a asexual reproductions. We cannot see any variations. Kandipa namakku endha variation paaka mudiyadhu illaya. All the plants are, so that is identical to the parents. Okay. And it is only one parent is enough for the vegetative propagations. Only one parent is required for the propagations. The offsprings are genetically identical to the parents. Okay. So what is the uses of that asexual reproduction is very, very important in the nursery and ornamental gardens, in the botanical gardens. It's very, very important because they have to increase their population within short period. Okay. That is undergoing the mitotic division and they have to spread over easily. Okay. In some plants, example for spinifex. The spinifex is here a type of grass. Okay. So this grass is, they can spread rapidly within the short period. Okay. So that is a spinifex. That is a type of vegetative reproductions. And next one, this is very important for the farmers and horticultures. Okay. They can use this method, they can harvest the plants in large scale. So in the mitotic divisions in the method we use pannumbodhu, so we can get it here a large scale of yielding. Okay. And what is the disadvantages? Only one disadvantage is we cannot see any new varieties. New plants are produced, have no genetic variations. Okay. So in each and every vegetative propagation, they can give rise to a new plant. But all the new plants are identical to their parents. We cannot see any variations. Okay. Any special characters we cannot see through this method. That is only one disadvantages. Okay. The advantages are very, very important for that. So board examination, usually they are asking this question. Okay. And next we are going to see the artificial methods. Okay. So artificial method means definitely we are going to do some artificial methods. Okay. Some of the artificial propagation methods have been used by man for continuous year in the long time, that is known as a conventional methods. Okay. But nowadays technology is being used for propagation to produce a number of plants within the short period. We can increase the population within the short period. So with the help of some technology. So what are the technology we are using nowadays? That is, we know very well, that is a tissue culture method or protoplasmic fusions or genetic engineering. Okay. With the help of this method we can increase the populations within the short period. So that is the modern method. Okay. So conventional method means the method is used by a man for a long time. Okay. That's a old method, is considered as a conventional method. So first we are going to see that conventional method. So the conventional methods are divided into three. One is the cutting, grafting and layering. Okay. So what is the cutting methods? So we can see the three types of the cutting. One is the root cutting, stem cutting and leaf cutting. It's a vegetative parts. Okay. Just you have to take the vegetative parts. It may be a root or stem or leaf. Just you have to cut it from the parental plant body, a small segments. Okay. So root cutting means just you have to take the long segments of the roots. Okay. Long segments of the roots. You just cut it from the parental plant body, and which is ability to form adventitious roots and buds. That is in the malus and lemon. Example for the plant, that's a malus and lemon. And next one is a stem cutting. So 20 to 30 centimeter long segments of the stem. That is, you have to take 20 to 30 centimeter. So cut it from the parental plant body. Okay. So actually this is induced root. Here we can apply some growth promoting hormones. Okay. With the help of the hormones, they can induce the roots very fast. So example for the hibiscus, bougainvillea and moringa. Okay, and leaf cutting already we discussed, there is a bryophyllum and begonia and scilla, we can use that. So for the leaf cutting, just you have to take a small segments. Already I explained in the begonia, just you have to take a damaged or injured leaves, okay, when that is contact with the soil, which gives rise to a entire plant body. Okay. And next method is a grafting. So what is the grafting? Grafting means that two different parts of the plant body is together, joined together. Okay, then they have to continue to grow as a single plant. Okay, rendu part eduthu enachtom appadina, so adu continue valarumbodhu ore plant adu maarudhu illaya, they have to grow as a single plant. So here we can see the some variations, we can see the characters of both plants. Enga irundha rendu part eduthamo and rendu part of the plants oda characters vandu nama anga paaka mudiyum. Okay, so there is a grafting. In this grafting, so what are the types we are going to see that? One is a bud grafting, tongue grafting, crown grafting, wedge grafting, and another one is your approach grafting. Okay, so what is the grafting? So definitions of the grafting. So here we are going to take the two different parts, okay, from the two different plants. So the both parts are joined, so they continue to grow as a one plant. Okay, and the two part of the plant body, so one is act as a stalk, another one is act as a scion. Okay, so which part is act as a stalk? So which one is contact with the soil? So that is act as a stalk. Edu vandu soil irukudho adha vandu stalk appdinu solrom. And another one is used for grafting, that is called a scion. Okay, so example for that one is citrus, mango and apple. So in these plants we can do the grafting method. You can see here, just they have to cut the plants, okay, from one plant body and which is joined with plant parts of another plant. Okay, so that is a grafting method. Grafting method is a common method. Even stem cutting method is also the widely used this method. Okay, so grafting is a common method. So next we are going to see the types of grafting. The first one is here bud grafting. Okay, so just you have to take a small bud like structure, okay, and the foliage buds and the bud region just you have to remove from the plant body. Okay, so the T-shaped insertion is made in the stalk. Okay, here T-shaped insertions, it means a small depressions. Okay, this is a T-shaped insertions made in the stalk. Edula T-shaped insertion kodu varano stalk la, okay va, and you have to see that this is here a bud. Okay, just take here a bud, foliage axillary buds vandu eduthukonum. So the bud is placed in the insertion regions and both of them is tied tightly with the any tape. Okay, so this is a bud grafting. Okay, just you have to take a bud from one plant body and keep it in that place of another plant body. Okay, that is a bud grafting. Okay, T-shaped insertion is mid insertion now, which is a depression mari edukrom. Okay, T-shaped insertion is we should take in the stalk. Okay, a scion bud abdingiradhu vandu, that is a take from the bud in the scion, another plant. Okay, so one is act as a stalk, another one is act as a scion. So placed in the insertion beneath and bark and properly, so that is a bandaged with a tape. So finally the both of them is joined together. Okay, so already we should take a small slice from both part of the plant body. Okay, the both of them is joined together and they act as a single plant. So we can see the different characters in the new plant. And next we are going to see the approach grafting. Okay, in the approach grafting we are going to take the two different parts of the plant body. So one is act as a stalk, that is a potted plant, that is act as a, so this is a stalk. Okay, and this is considered as a scion. Okay, this is a stalk, this is a scion. Okay, the potted plant should be brought closely to that stalk, that is a scion plant. Scion plants ku vandu close ah nama adha kondu varum. Okay, so we have to take this stalk is grown in a part and it is brought close to the scion. Okay, potted plant eduthu abbi scion plant, scion part edu, that is a tree is a scion part. Okay, so the close edutittu vandu and the both of them is tied together and before we must take a small slice from both regions. Okay, so actually we should maintain the same thickness of the branches. Rendulayum same thickness of the branches vandu nama first select panikanum. Okay, so that is a slice, a small slice should take from the both branches. Okay, then only the both of them is fused together. Okay, so after the 2 to 3 weeks, just you have to make a cut in the base of the scion, and that is the apex of the stalk. So this is a stalk, this is a scion. Yeah, so we have to cut it in this region. So this portion, the potted plant, is separated from the process. So it will grow as a new plant, and they can give into your new characters. Okay, so same thickness should be maintained in the approach grafting. And next is here tongue grafting. Tongue grafting, here also we should maintain the same thickness. Okay, so here we have to cut the scion and stalk in obliquely. Obliquely அப்படிங்கிறது ஒரு slanting positionல நம்ம cut பண்றோம். Both of them is cut in the same positions, and both of them are maintaining the same thickness. Okay, and the stalk and scion is, they have to fuse it to one another and tied with a proper bandage. Okay, so after a week it will grow as here, after a month or after a week it start from the fused in the after a week. So after 1 or 2 months, both of them is close to each other. Okay, fused to each other, they act as a single plant. That's a tongue grafting. So what is we are going to maintain in the tongue grafting? Same thickness, and the cutting is obliquely cutting. And next one is a crown grafting. Crown grafting, here we are taking the stalk is enlarged in size. The stalk pathina not same thickness. Okay, the stalk is your bigger size. Stalk vandu romba enlarged flat ah irukum. Okay va, and that scion is, that is your thinner. Actually we have to take a wedge shaped structure. Okay, so this is a wedge shaped structure. We can do so many scions, we can insert so many scions inside that in the stalk. Just we have to take a slits or clefts. Slits அப்படிங்கிறது ஒரு சின்ன so hole மாதிரி ஏற்படுத்தலாம், இல்லைன்னா clefts. Just we have to take a clefts. Okay, inside the clefts we have to insert the scions. Okay, 1 or 2 scions. Okay, more than 1 scions we can insert in the crown grafting. And next one is a wedge grafting. Okay, so in the wedge grafting, so the both stalk and scion in the same thickness. Okay, in the scion should be wedge shape, but the stalk should be, that is your V shape. இதல பாத்தீங்கன்னா உங்களுக்கு clear-ஆ தெரியும். So this is your stalk. The stalk should be V shape. The stalk வந்து எப்படி இருக்குன்னா V shape ல இருக்கு. இதுல இருக்கக்கூடிய அந்த scion வந்து எப்படி இருக்கும்னா wedge shape ல வந்து இருக்கும். Okay va? So that is, both of them is fused together. Okay, here both cambium of the two plants are joined together. Okay, rendu cambium, because we have to remove the bark regions, and that is here in cortex, endodermis, and the மேல இருக்கக்கூடிய எல்லா regionையும் remove பண்ணிட்டு, so that's a cambium of both plants are joined together. And next one is here layering method. So layering method is a very common method, we know that. Okay, so in the layering, just we have to take a plant. Okay, that is a flexible branches of the plant, we should select it. Okay, the flexible branches that is here buried in the soil, from the branches of the stem, that's the roots are raised. Okay, so in this method the stem of the parent plant is allowed to develop roots while still which is attached to the parental plant. Okay, so parental plant oda irukumbodhe idhula irundhu enna agudhu, roots vandu raised agudhu. So here we are going to take the flexible branches. Okay, the lower branches of the plant is just, they have to bend down in the soil. Okay, the part is buried in the soil. So after that is 1 or 2 months, we can see the roots are raised from the, so that is a stem that is buried in the regions. Okay, just you have to remove the part from the plant body which is act as your, so new organism which is gives rise to your entire organisms. So here that Ixora and Jasminum, we can, Ixora and Jasminum is the best for that layering method. So Ixora, Jasminum. And next, mount layering method. Okay, so in the mount layering method, this is just what I explained in the layering, that is the same thing is here. Here the plants having the flexible branches, we have to take, okay, the flexible branches of the lower stem. Okay, so that is buried in the soil. Okay, that is covered with some mud. Okay, full total covered pannirunga. So after 2 to 4 months or after the 3 months, we can see the that is the roots that is emerged from the buried regions. That is, you have to cut it from the plant body and keep it in some other places. That is a mount layering. But here that is your aerial portions, that is the leaves portion should be, there is exposed to the, exposed above the soil. Okay, so on the branches mattum ulla buried pannirkom, the aerial parts vandu it should be exposed into the, so soil. And next one is a air layering. So air layering is a very best method. So here we can bring some variations. So in this method, the stem is griddled. It means, so we have to take a 3 to 5 centimeters. The ring of bark should be removed. Okay, in this region, so 3 to 5 centimeters, a bark should be removed. Okay, that's a griddled. Then this region is covered with a thick plasters of, that is a grafting clay. Okay, grafting clay means it consists of a clay and that is some other humus soil that is mixed together with. Okay, and another one the cow dung, and we have to add a little quantity of the hormones. Okay, so what type of hormones we have to add? That is a growth promoting hormones. So that's a NAA is a growth promoting hormone. Naphthalene acetic acid is a growth promoting hormone. Adhe mari vandu indole butyric acid is a growth promoting hormone. Just you have to add little quantities on these regions. Okay, and everything is covered with this region, covered with the polythene. Okay, so the total region is covered with polythene. So after 2 to 4 months, we can see the roots are emerged from these branches. Okay, then we have to cut the branches and keep it in some other places. It will give rise to an entire plant body, and we can see that some variations. Okay, so other variations due to the environmental changes, we can see some variation. The plant should be very healthy because already we added some humus and growth hormone also. Okay. So what are the advantages of the conventional method? Okay, conventional method is the old method. So this is also similar to that vegetative propagation. That's the plants produce genetically uniform plants. So many plants can be produced quickly by this method. It looks like a tissue culture method. It will be later. Okay, quickly we can produce the populations, and some plants produce little or no seeds. Okay, some plants seeds very less. Some plants seeds irukadu. Okay, in this condition we can use this method. And another one, that is some plants can be propagated more economically by vegetative propagation. That's an example of Solanum tuberosum. Okay, Solanum tuberosum everyone is cultivating in the Bengal. Okay, so they are cultivating the Solanum tuberosum. It's economically valued. The two different plants with desirable characters such as disease resistant, high yield can be grafted. So that is a grown new plant with the same desirable characters. If you are using the desirable character of the plant body, so we can get it the desirable characters in the new plant body. Okay, so the desirable characters desirable characters and what are the disadvantages? Example for we are not at all using the desirable characters. So unfortunately, if any diseases in the plant body, definitely it will be spread over to the next generations. The use of virus infected plants as parents produce the viral infected new plants. If virus irunduchuna aduthu varakudiya thalamuraiyilayum adha virus vandu irukum. Okay, so vegetative structures used for the propagations are bulky, and so they are difficult to handle and store. Okay, example for sugar cane. Okay, the stems are vegetatively propagated, isn't it? So adu nariya irukum, adha nama store panni vekiradhu, sila propagated vegetative propagated materials store panni vekiradhu is not easy. We need more space for that. Okay, this is the disadvantages. Okay children, I hope you could understand this concept very easily. So thank you so much for your cooperations. So really, if you like this video, please like it, share it and subscribe.